Welcome to Health Matters, where we explore the science and stories behind the health issues that matter most to you. Today, we're exploring the fascinating world of gut health with Dr. Goodnow, a renowned biochemist with an in-depth understanding of how our gut is biochemically linked to the rest of our body. Dr. Goodnow's research extends beyond brain health, encompassing large-scale clinical trials and the development of screening tests for colon, pancreatic, and liver cancers. Maintaining a healthy gut is crucial for overall well-being, as it plays a key role in digestion, metabolism, and immune function. When gut health is compromised, it can contribute to various health issues, including inflammatory bowel disease, irritable bowel syndrome, allergies, and even mental health disorders.
We think about the bacteria, it's a two-way street. Your gut microbiome is adaptive to your biochemistry. So if you have type 2 diabetes, if you have stress, if you have certain things in your life, your bacteria will adjust to it as well, because it'll sense the blood supply, your metabolites in your blood, because your blood supply is still connected to your gut. And likewise, your gut microbiome will interact. So it's a two-way street on these gut microbiome. So that's how it happens. That's how it's supposed to work.
In today's episode, we'll explore the gut's role as a key player in digestion and how we break down the food we eat and absorb essential nutrients. We'll discuss the microbiome, a diverse community of trillions of microorganisms, and its critical role in maintaining gut health and preventing inflammation. Dr. Goodnow will also shed light on the gut-brain connection and how the health of our brain is deeply intertwined with the health of our gut. We'll also discuss how your blood work may even be able to reveal insight into the state of your gut health. So, whether you're curious about how your gut works, what happens when you eat, or how to maintain a healthy microbiome, this episode is packed with insights that reveal how essential gut health is for your overall health. Let's get started. It's time for Health Matters. Hey, Dr. Goodnow.
Hi, Victoria.
How's it going?
Very good. Exciting time today.
Yeah, well, you never cease to amaze me because, I think of you as someone who knows, obviously, a lot about the brain and biochemistry. Right. And when I was looking more into your background, you have a lot of experience in gut health as well, whether it's large-scale clinical trials for, was it colon cancer? Tell me a little bit about the work that you've done in the gut health world.
Yeah, the whole gut health, colon cancer, pancreatic cancer, esophageal cancer, stomach cancer, and so on, really came by organically. As you know, my formal training is in synthetic organic chemistry. Mm-hmm. My PhD is in psychiatric medicine, looking at the biochemistry of neurological disorders. And then in the late 90s, I invented this technology called non-targeted metabolomics using ion cyclotron mass spectrometry.
Which is basically looking at the biomarkers of blood.
It is comprehensive, exactly. It's ability to basically measure everything in blood and how it relates to human health. And we also use this technology in functional genomics and other preclinical research models. And the technology was so powerful in identifying changes with physiology, right? And so first you think about Alzheimer's and Parkinson's and things. But the other major diseases that really affect the biochemistry of health, as you would imagine, would be the cancers, right? So, and those are the ones that we really, from a population perspective, we really want to be able to screen for colon cancer. Can we identify people with colon cancer early or at risk of colon cancer early? Or pancreatic cancer is a big one because it's one of the mortality rate of pancreatic cancer is so high, like it's 85, 90%.
Wow.
Whereas colon cancer is, you have a much better chance of surviving colon cancer.
And that's why they implement screenings where, a certain age and they're kind of moving the screenings back and back saying, oh, you might need to get a screening at a younger age.
Correct. And that's exactly right. So the issue then is, how do you identify these high-risk diseases early? Because everyone knows if you can treat colon cancer, pancreatic cancer, or any cancer earlier, your survival rates are much higher. And so this metabolic technology was ideally suited to try to understand the basis of health and deviations from health. And so the logical way to apply this was in the field of certain cancers, especially those cancers in which we have ineffective screening technologies. Even colon cancer, for example, we use a fecal immunohistochemical blood test, which basically looks just for human hemoglobin in feces. It's a very non-specific test. And they've tried different genetic tests for colon cancer screening, but they take a big stool sample to work. And then pancreatic cancer is another big issue, is that the incidence rate is low, so you can't really screen everybody for ultrasounds and so on. So there's really need for early markers of cancer or risk of cancer. And ovarian cancer is another big situation because it transitions from stage 1 to stage 3 so quickly when it goes through the serous model. So using this technology really was understanding a lot of work on cancer. And being a biochemist, biochemistry of the brain, biochemistry of the gut, biochemistry of the liver is still biochemistry. Like all these systems have the same core operational components of it. And ultimately, when we're talking about restorative neurology or restorative biochemistry, which we do a lot of, well, you can't really do restorative biochemistry if you can't understand how to get nutrients into the human body and into the brain or into the heart or into the lungs for the people. So understanding the gut is a core component of all types of pharmacokinetic research. Like we're saying, okay, if I want to restore lung function, well, how do I get something from the food supply to your lungs? Well, it goes through the gut. It goes through the absorption, digestion process, metabolism, excretion, like classic ADME type technologies. So that's a core component of all aspects of clinical design of nutrients, as well as clinical design of therapeutics. So that's why I got naturally into the gut world. And quite frankly, it was colon cancer that was the biggest eye-opener for us, or for me, understanding the deviations from health versus the diagnosis of disease.
Which you call prodromes, right?
Prodromes, right.
The things that come before, the writing on the wall that says this is imminent because these things are out of balance.
Exactly. And so you think, so the logic at the time, back in the 2000s, was, well, if someone has colon cancer, clearly they've got to have biochemical markers of the colon cancer, right? We're thinking, okay, can I find biomarkers of colon cancer in the blood? And so we use this advanced ion cyclotron technology to measure the comprehensive metabolome of the blood in individuals with colon cancer versus people without colon cancer. And lo and behold, we found dramatic biochemical changes. So people with colon cancer have a dramatically altered biochemical profile in their blood, as you'd expect, right? But the hypothesis was that, the cancer is creating all of these abnormal biochemicals. So we're going to see these cancer biochemistry in the blood. And what we found was the opposite. We found that there was a deficiency of certain molecules in the gut, in the blood, coming from the gut. And at the time we thought, well, then if we cure the cancer, like go through surgery and someone gets technically reported as being cancer-free, then these biomarkers should come back to normal levels. Well, we found that didn't happen. What happened was that the biomarkers stayed the same even after the cancer was removed, and the person was technically considered cancer-free. And we didn't even believe it at the time, so we redid the entire clinical trial over again with a different academic institution in Osaka, Japan, and we got exactly the same results. And so this was so in many ways, colon cancer is what really created really solidified this concept of prodromes, because it became very clear that there's a biochemical change that occurs before cancer. And what was interesting about these biomarkers that were decreased in individuals with a high risk for colon cancer was that these are molecules that your body doesn't make, but your gut microbiome makes. And so it clearly indicates that when you have a dysfunctional gut microbiome, that is actually a high indicator of gut health, but also cancer risk in situation of colon cancer. So that's where biochemistry comes in, because your gut is a huge biochemical engine, if you will, or bioreactor. Just like, making beer and wine, you have this whole, process.
Yeah, that's so interesting what you said in the beginning about how, I guess I never really thought about it so simply, but we are, bioreactors, and it doesn't matter how healthy your food is or how impressive your supplements are if you can't process them.
Correct.
And so two people could be probably eating the same things or taking the same protocols and have different success because of how their gut is functioning and how they're able to process what they're putting in and get it to where it needs to go in the body.
Correct.
That's so interesting. So when you mentioned with the colon cancer screening or
Yeah.
this trial you were speaking about. You took the cancer out, but the blood work, the biomarkers stayed the same.
Correct.
And then you tried it again, and the same thing happened.
Correct.
So that means that there is a prodrome of all. When these things are out of balance, it will lead to. It could lead to like colon cancer, for example.
Exactly.
So how do you fix the biomarkers?
So the biomarker, that's obviously that's the amazing question, right? So in terms of the gut microbiome and how it gets dysfunctional with different diseases, antibiotic use, diet, exercise, 100 different things can affect your gut microbiome, right? Then that sets you up for future risks because the gut microbiome is highly integrated with your own human biochemistry. So we should probably start backwards a little bit and saying, where does this all come from in terms of when people think about the gut, anything and everything that gets into the human body comes in through one of a relatively small number of areas. You can get absorbed through your skin, for example, okay? Or you can breathe it into your lungs. But fundamentally, most things go through our mouth, right? And so whatever gets into our body usually has to get in through our gut. And whatever gets blocked from coming in gets blocked from our gut. So fundamentally, since we are these bioreactors, whether we get healthy nutrients from our food supply or we get toxic nutrients from our food supply is fundamentally driven by our gut function.
Wow.
And our gut, and people think your gut's inside you, but it's really not. It's like this tube that goes from your mouth to your butt, okay? And it's like your skin folded in on itself because, you can take a thread and you can run it from your mouth all the way out your butt, and that's actually not technically in your body, right? Like that's actually, it's actually outside of your body, but inside at the same time.
Well,
it's kind of weird. But
it's in your body in the sense that it's like inside of you, but you're saying it's not like in your blood. Like it's like a closed system where it's in the pipes, but it's not like in your blood or circulating around you. Is that what you mean?
Exactly. And so, and we're aerobic organisms. So all of we breathe air, which has oxygen in it. And then we oxygenate, all of our blood system has oxygen permeating through it and carbon dioxide leaving. Your gut is fundamentally anaerobic. It's like oxygen. So the microbiology of your gut microbiome, 99% of those microbes are called anaerobic microbes. So they do not use oxygen.
Versus the ones, the microbes in your mouth are able to withstand oxygen because there's a lot of oxygen obviously coming in through your mouth.
Correct.
So when we talk about gut, it's this whole system that includes your mouth, your, I guess, esophagus, stomach, pancreas.
Yeah, well then the
liver,
the small intestine down to your large intestine and to your colon. And then you have these organs that are connected to your gut. Okay, like your liver is connected to your gut.
Okay.
Your pancreas is connected to your gut. And these provide enzymes and hormones back and forth from the gut, back and forth from these organs in terms of digestion and processing.
So walk me through like what happens when we eat, from like the journey of a, bite of salad. Yeah, exactly. Hamburger. I was going to say salad. I don't know why.
How does a hamburger
so
get
boring.
protein and carbos and sugar into your body? So fundamentally how the gut's supposed to work, okay, is that obviously you have your mouth. And so we have a certain amount of chewing that can get done. And there's a certain amount of absorption of simple nutrients. So most people think, like when you look at the nutrient list on your quart of milk, right? It always says how much fat, how much protein, how much carbohydrate. So these are the three main food groups, if you will. And then we have all these phytochemicals like people talk about, right? Like curcuminoids and things that are not human, but we are healthy for us in our diet supply. But the three main things that humans live on are carbohydrates, fat, and proteins. And we get those from food supply. And so the primary objective of the gut system is your enzymatic digestion of those simple core building blocks. And so in your mouth, you'll have certain enzymes like amylase and some proteases that will break down sugars quite quickly. And you can absorb some of that right in your mouth. Like people talk about sublingual absorption of, drugs. Like many people have a sublingual melatonin, for
Mhm.
example.
So
And
those bypass the stomach acid?
Correct.
Okay.
They could get absorbed through your mouth walls. But normally when you're eating a food, it's somewhat complex, right, at first. So it's not digested. It's still in complex proteins, complex. Like you don't absorb a protein. You don't absorb carbohydrates. You don't absorb fat directly. It has to get digested first before you absorb it. So then it goes from your mouth down your esophageal cavity into your stomach. And there it experiences what's concentrated hydrochloric acid. And you'll notice that if you smell pickles or something and you get a salivary response, you can actually stimulate stomach acid formation. And so
Pickles.
exactly, whatever causes you to have this smell of you can anticipate some food product, right? And that again shows you how connected your brain is to your gut. Just the thought of thinking about pickles can cause you to actually secrete enzymes and acids into your stomach. That's how powerful the gut-brain connection really is, because we can think about food and we can neurologically stimulate our gut without having any food in our system, right?
Or even like they say that there's obviously science to a gut feeling. Like you can think about something and feel it in your gut as well. So
and we shut our gut off and on based upon stress responses as well, right?
Mm-hmm.
And so there's a highly connected aspect. So anyway, so it gets through the first part, it gets into your stomach, and that's where it gets exposed to concentrated hydrochloric acid. Stuff stuff will take paint off. Like it's really, really, so it's amazing that your stomach walls, the lumen of your stomach, can tolerate not a bucket, but a cup of concentrated hydrochloric acid. And so that's actually the first protective barrier to a lot of bacterial infections, is that acid in your stomach. And it also helps start to digest and start to loosen up certain molecules, especially fats, for example. And so your stomach will have some enzymes called gastric lipases, because the first thing the body likes to absorb is fat, actually, because fat's a critical energy source. If you're starving in the middle of the desert and you get some oil, fat, like olive oil, your body will absorb it very quickly and get it to your muscles and your heart and your brain. So anyways, gastric lipases come in. So your stomach actually secretes enzymes that are designed to digest fat or triglycerides. And it digests them into these called free fatty acids and monoacylglycerols. That's what gets absorbed. So when you take olive oil out of your cupboard and you drink it, olive oil does not get absorbed into your blood supply. Olive oil gets digested by what's called lipases into free fatty acids, monoalkylglycerols, and that gets absorbed through the lumen walls, and then that's the blood supply, and then it takes those molecules and reattaches them into triglycerides into your liver.
What are the lumen walls? That's the walls of your stomach?
Yeah.
Okay.
And lumen, either the walls of your stomach or the walls of your intestine.
Okay. So it takes the oil, breaks it down into things that your body can use, these free fatty acids, and then it is absorbed from the walls of whether it's the stomach or intestines into the body.
Correct.
Okay.
Yeah, and so the enterocytes, they excrete mucus, so there's a mucus membrane. So technically, the lumen is the center of the tube, if you will. The outer walls are the enteral mucus coatings of your and your enterocytes excrete these mucus, and so you have different types of microbiomes. Like your microbiome of your gut changes from your mouth to your butt, also changes from the outside walls of your intestines to the inside.
Wow.
So you have different types of bacteria that are in the mucus lining of your gut versus the center, if you will, of your gut flow. Anyway, so back to
so from the stomach, where does it go?
From the path here. So once the stomach gets full for a certain while, there's a certain time called gastric emptying rate, and then it'll pass through your pyloric valve into your upper small intestine. And now a second set of enzymes come in, like pancreatic enzymes, like pancreatic lipase, which is additional for fat. There'll be proteases that'll come in and start metabolizing some of the amino acids. So proteins are these complex proteins. So people have allergies for certain proteins, for example. Your gut is supposed to digest those proteins into, free amino acids. And those free amino acids get absorbed and into the body. So that's why the gut basically takes this milkshake and Big Mac burger, and actually digests it into small components.
So what about the sugar? Like everyone talks about glucose and glucose spikes and all these things.
Yeah.
When does, I guess, insulin come into play? Is that in the small intestine?
Well, it's stimulated. Insulin's from the pancreas.
Okay.
But it comes from your blood supply once it gets absorbed. So simple carbohydrates, like when you take a starchy food, like a potato, so complex carbohydrates are chains of glucose molecules. And when you talk about diabetes, when you measure your glucose in your blood, so glucose is the fundamental sugar of human biochemistry. When we talk about fruit sugar, which is called fructose, or when you take a table sugar, which we typically call is usually called sucrose, and sucrose is one molecule of fructose and one molecule of glucose together. So anyways, different types of sugars based upon how the glucose molecules are connected to each other can be either quickly digested or slowly digested. And so sucrose is actually not that bad of a sugar. What's really bad is like pure starchy type foods because your body will break that down quickly. So it takes this very complex chain of hundreds or thousands of glucose molecules, and it breaks them down to single glucose molecules. And those single glucose molecules then get absorbed into your blood supply. And when that goes into your blood supply, your blood glucose levels go up. And when the blood glucose levels go up, it signals to the pancreas that, hey, you're eating, you have high sugar. And then your pancreas inlet cells will start releasing the protein insulin. And so the insulin goes and tells all your body cells to start sucking up glucose to bring the glucose out of the blood supply into your cells.
Insulin is a protein?
Yeah. So protein, it's a peptide type hormone that signals cells to start taking up glucose. And when it takes up the glucose, it starts converting the glucose into glycogen, which is a storage molecule, or converting it into triacylglycerols.
So when you eat a bunch of sugar, when you eat a bunch of sugar, your pancreas sends out insulin to absorb the glucose or send it to different places where it needs to go?
Well, it sends it to all your cells of your body. So all your cells of your body start sucking up glucose to get the glucose out of the blood and bring the glucose levels down. Because if you have high sustained blood glucose levels, it's very dangerous. It's blind. You go blind. You can.
What does it look like for someone with type 1 and type 2 diabetes, this process?
So type 1 versus type 2. So type 1 diabetes is a disease where there's an actual, the inlet cells, the insulin producing cells of the pancreas become damaged.
Okay.
Typically it's an antibody, it's an autoimmune antibody response that occurs. And so then what happens is people with type 1 diabetes, they don't have any insulin signaling response. So when blood glucose goes up, the pancreas can't produce insulin. So those individuals, they need insulin all the time because their pancreas can't actually produce insulin. So that's type 1. Type 2 diabetes is called insulin insensitivity, which means that when you take a big meal and your blood glucose levels go up, and then your pancreas is being stimulated to produce insulin, it produces insulin, but the insulin that it produces doesn't lower blood sugar levels enough. And so when it's signaling to your cells to say, hey, suck up glucose, the cells are saying, sorry, I'm all full. I can't take any more glucose. And so people are then considered glucose insensitive, insulin insensitive. So then you need more insulin. More insulin is required to lower blood glucose levels. So that's why people with type 2 diabetes, they will get an additional insulin injection, which will supplement their current pancreatic release to try to lower blood glucose levels.
I've never really thought about how diabetes is actually, it's just like a metabolic insufficiency,
Correct.
Right? Like you're unable to process energy.
Right. So type 2 diabetes, okay, is basically glycolytic insufficiencies. So the cells of the body cannot process glucose efficiently. And so all the things that cure type 2 diabetes, which is resistance training, exercise, lower glycemic index food processing, all those things, they work because what they do is they improve the glycolytic capacity of your cells. So it allows those cells to actually do the job that insulin is asking it to do. And so what's happening in classic type 2 diabetes is insulin is asking the cells to do something that they cannot do. And so that's why we have type 2 diabetes rampant. And it happens as we get older when we lose our muscle mass, because our muscle mass is critically
So that takes a lot of glucose, right? Or your muscles can...
Yes, they absorb a lot of food energy, basically, fatty acids and glucose. So anyway, so then as it's being absorbed, but then of course your gut microbiome is sensitive to certain types of food supplies, right? So when we talk about, so as it goes through your gut, so that's your basic food groups, right? Okay, but we don't eat nice, clean, pure carbohydrates, fats, and proteins, right? We eat a whole bunch of stuff in our food supply. So the gut microbiome becomes a symbiotic relationship with the body. And so it's growing in our body. It's using glucose in an anaerobic metabolism perspective. So it's using some of the energy that we are eating. And it's using that energy because those cells are alive, right? And those cells are alive so they can actually metabolize things. So when people take fiber in their diet, for example, say, oh, I need fiber because fiber creates butyrate. And so like when your gut microbiome metabolizes fiber, it will create a molecule called butyrate, which is healthy for you. It's a stimulating molecule for mitochondrial function, or acetate is another good one, like that kind of thing. So anyway, so your gut microbiome, but also your gut microbiome does a couple other things. One, it will digest, it'll have enzymes and it'll have metabolic systems that we don't have in our body. So the microorganisms will have certain biochemical capacity that a human cell, like your liver cell, can't process. Like it'll have enzymes that your liver cell does not have. So it can actually metabolize in certain molecules that the human body cannot metabolize. And so it can make those molecules actually biologically available. So we call those things sometimes like prebiotics. So take a molecule or a sugar called lactulose, like we use as a prebiotic. The human body can't metabolize lactulose, but enzymes in the gut microbiome can.
So you're not feeding your cells. You're feeding the microbiota that are living in your body.
And then they're creating molecules that are being absorbed. So this colon cancer and pancreatic cancer biomarkers that we talked about, right? I call these gastrointestinal tract acids, GTAs. And they're very long chain dicarboxylic acids. They range from 26 carbons to 36 carbons. And they're all deficient in the blood of individuals with colon cancer, pancreatic cancer, stomach cancer, and even ovarian cancer.
Wow.
And so these are molecules that the human body does not make. Like our biochemistry does not make them, but our gut microbiome does make them. And these molecules have extreme anti-inflammatory activity, anti-cancer activities. They enter the human brain because they're found in, we can detect them in the human cerebral spinal fluid. And so here's an example when the gut microbiome is not producing molecules that are helpful to humans. So the gut microbiome is actually preventing people with a healthy gut microbiome. It's that microbiome that's actually giving them cancer preventative capacity by these molecules.
It's really interesting. Like the microbiome or the bugs in there are also sometimes the ones who are craving the sugar. Like not even like you. Like there's some really interesting studies that took taste out of it. Like they were able to just kind of put the food directly into someone, and whether they're not even tasting the sugar, but they're craving more of it because the bugs in their gut are craving sugar.
Right.
And so sometimes when you have these horrible cravings, it's actually your microbiome, then these little bugs saying, give me more sugar, give me more sugar. So you want to be in balance. It's not like you defeating the microbiome definitely helps you. It helps your immune system, helps you process food. Like the healthier your gut is in this microbiome that's living within you, the better. But, it can kind of, some of the bad bugs can win if you don't have like a balanced diet.
Correct. Well, it's a diversity. Okay. And it's a community. Like the gut microbiome is a community of cells. And so a diverse gut microbiome is a good one because it actually can handle multiple different types of insults. It's like having a diverse immune system, right? Like if you were born and you just lived in a bubble all your life and then you get exposed to the environment, okay, you're going to get sick because you've built up no immunity, you have no immune system, you have no antibodies to common pathogens and so on and so forth. Your gut microbiome was in a very similar way. You should have a diverse gut microbiome that can handle a variety of types of environmental insults that may or may not happen. And it's dynamic. It will adjust to your environment. And this is where the prebiotics come in. So actually, high sugar is a prebiotic for bad bacteria, right?
So prebiotics are the food for the microbiome or the microbiota.
And you're kind of selectively, you're selectively feeding, because all these microbes, they'll have slightly different tolerances, slightly different aptitudes, if you will. And so that's where lactulose is an interesting one. So lactulose is a simple sugar that has.
Can you buy it?
Yeah, it's usually by prescription, actually.
Oh, okay.
It's for use for diarrhea, for constipation and so on. But it also is being used more frequently now at low doses for a prebiotic, because two of your really healthy gut bacteria, called your bifidobacteria and your lactobacillus, it happens that these two types of healthy protective bacteria, they're two that they can more efficiently metabolize lactulose than other bacteria. So if you take lactulose, it's going to preferentially support bifida and lactobacillus, and they will grow to larger populations because you're feeding them selectively. Whereas if you provide pure sugar, glucose in your diet, then yeast and other things that are really high glucose sensitive or glucose loving bacteria, if you will, they will populate. And so when people talk about the gut dysbiosis, is usually when one type of bacteria overtakes everything else, like C. difficile, and some of these bacteria then will create toxic molecules. So back to this whole microbiome, it's very complex, but it's actually very interesting in its complexity, because our gut microbiome is adapted to humans. Whereas a cow and a cat and a dog, they'll have different microbiomes. The microbiome of the soil will be different. And those, or microbiome that coexist, say, with legumes that that capture atmospheric nitrogen and converts it into, soluble nitrogen, so on and so forth. So the symbiotic relationship with the bacteria in our universe, they kind of adjust and become symbiotic. Now, it doesn't mean you can get a bad bacteria. We talk about biology, right? When someone says you get an invasive fish in your ecosystem, so someone takes the wrong fish, or you say, okay, make sure you clean your boat off and get all the seaweed off your boat, because you don't want an invasive plant species going into your lake. Well, that's the same concept, right? So if you bring in a species that doesn't have the appropriate checks and balances on it, then it will proliferate in excess of its boundaries. So the same thing can happen in the human gut. So you can get a bad bacterial infection, right? So you can get bad bugs, or you can take antibiotics that will selectively kill certain bugs, and you won't get repopulated. So that's why once you actually have a healthy gut microbiome, it's pretty resistant. Like once you have a healthy gut, it's pretty hard to destroy it. Because it's quite adaptable, and it'll come back. And this is why, right from birth and early breastfeeding really affects our entire gut microbiome. So actually a vaginal birth is different than a cesarean birth. So when a baby is born and it goes through the vaginal cavity, the baby gets exposed to the mom's bacteria, like the mom's microbiome. So there's a vaginal microbiome, just like anything else. And that means the very first, inoculation of the baby is going to be similar to the mom. Now, C-section, okay, so there's differences between how the bacteria become populated. That way, same thing with breastfeeding versus non-breastfeeding. So one of the things, and you can buy these probiotics, like one of them is Lactobacillus reuteri, for example. There's a product like Gerber Soothe, or there's other brands of it. And that's one of the first bacteria that gets passed on to the baby through breastfeeding. Okay, so when a baby gets breastfed, they're going to get exposed to certain bacteria. And so they're getting probiotics from the mom, right? And so people think about, and so the trick for a healthy bacteria is to try to get that base bacteria and then maintain it. And then the final equation of this situation is that we think about the bacteria, it's a two-way street. Your gut microbiome is adaptive to your biochemistry. So if you have type 2 diabetes, if you have stress, if you have certain things in your life, your bacteria will adjust to it as well, because it'll sense the blood supply, your metabolites in your blood, because your blood supply is still connected to your gut. And likewise, your gut microbiome will interact. So it's a two-way street on these on the gut microbiome. So anyways, so that's how it happens. That's how it's supposed to work. So then what your gut microbiome does is it's there. It provides you, if you have a good microbiome, it's providing you with healthy molecules that actually enhance human health. Okay, they provide us with things that we can't make ourselves. That's on the positive side. On the negative side, okay, if you have the bad bacteria, or if you don't have the right bacteria when you get exposed to certain food products, a good example is food sensitivities, right? So one of the biggest issues with gluten sensitivity, or people that say, I can't eat at restaurants, they use ice in their drinks and gives them diarrhea, that kind of stuff. That means that their gut microbiome can't process these varieties of environmental foods.
Or even when you travel, right? Because you're in a different ecosystem with different bugs, and you're drinking the water, eating the food that's different. And if you're not adjusted to it, your own microbiome that has been cultivated over your whole lifetime is not ever had to experience those things. Yeah.
And so people get really amazed all of a sudden when you start actually correcting the gut microbiome, and they start realizing, oh, I can eat foods that I used to not be able to eat. It's because your gut microbiome has now been able to become more adaptable and more resilient to a variety of environmental exposures.
And that's why at a young age, like you mentioned, C-section birth versus how you're born, that's exposure to, microbiome or a different host of things.
Right.
But also I was even reading that having a pet at home as a kid, how many caregivers are interacting with you versus is it the same person? If you're playing outside, all of these different things, it's just like exposure and diversity.
Correct.
That helps to build the immune system and the microbiome. I thought it was really fascinating.
It really is quite amazing. And so this obsession with, antibacterial soaps and everything else, you have to be careful with that kind of stuff, because it can actually disrupt.
It's like a shotgun approach where it kills everything, even the good stuff. Something you'd mentioned earlier, I think it was bifidobacteria. Yeah. Back when I worked in the neurology clinic and we were looking through patients with, neurodegenerative diseases, I found it so fascinating that a lot of them had almost no bifidobacteria. So, there's got to be this gut-brain connection that everyone speaks of is obviously very profound. Can you talk more about this gut-brain connection? How are they actually connected?
Yeah. So first of all, when we talk about the gut and the gut-brain connection and gut inflammation, for example, right? So we have the bacterial distribution from right next to the enterocyte wall into the center of the lumen. And so you have someone with colitis, for example, like their gut walls are inflamed, right? They're red. And that means that the bacteria in the gut are actually emitting pro-inflammatory molecules. And your gut wall is actually reacting to that inflammation. And so, it's a self-perpetuating loop, because the inflammation, then you would think it doesn't follow the regular rule that you become tolerant. Like, so, you become resistant to something after a while, right? What happens in, say, something like colitis is that the fact that you have inflammation of the gut lining, there's only a certain type of bacteria that will live in that environment. And so it actually promotes itself. So it actually protects the type of bacteria that like to live in inflamed gut.
But then how do they get to the brain, or how do they interact with the brain?
So now when you talk about the direct connections, like how we feel with our gut and so on and so forth. So the lack of bifidobacteria is a big deal, because it's a very protective bacteria. Also, your bacteria is somewhat of a zero-sum game, right? Like, you have a certain volume, and it is either you have 10% of this and 80% of that, or you have 50-50. So it always adds up to 1 in a sense. Like if you have 30 trillion bacterial cells in your gut, you kind of always have somewhere in that 30 trillion cells. The question isn't how much bacteria you have, it's the distribution of bacteria.
It's the balance.
So if you have low bifida, by definition, you're going to have high something else. Okay. And that high something else is unlikely to be as good as the bifida. But back to the neurological connections. So the gut is a very large organ, right? And it is connected all the way from your mouth, your upper intestine, to your colon. And it is controlled neurologically. So you have the vagus nerve, which comes from there's basically 3 types of neurological connections with the gut. There is what's called the efferent, which is from the brain to the gut, which is typically like the vagus nerve. And that's usually a constrictive response. It's the vagus nerve, which releases noradrenaline. So the brain can quickly squeeze your gut, shut your gut, right? You make a joke of, so if you're in a fight or flight response, it just shuts your gut right down, and you can redistribute it. Your vagus nerve also allows you to redistribute your blood flow from your heart and lungs to your gut or to your kidneys. So the redistribution of your blood flow, because your gut, when you take a meal, right, and if you're nice and quiet and you just finished watching the football game, and your gut will say, your body say, oh, okay, we're in quiet mode here. Let's put a lot of blood to the gut. Let the gut just absorb. We're gonna focus on all the energy required to digest all this food. Okay, but if you eat a big meal and all of a sudden some guy comes in with a gun, okay, you're not digesting any food anytime soon. Okay, you're shutting down and you're in a fight.
Yeah.
So that's all neurological. So that's the efferent. That's the ability of the brain to actually control your gut based upon dynamic situations. Then the afferent is what we call from the gut to the brain, where the brain is sensing the gut. Like, am I full? Has there been some issues? But those are actual neurological connections. Like, so when we talk about the blood supply, people think, oh, there's a blood-brain barrier. Well, there's no such thing as a gut-brain barrier. Okay.
Wow.
So the neuron that goes from the cell body, if it's the efferent, the cell body is in the brain cavity, if you will. And then the axon is going all the way from the brain to the gut. And so that is one connection. There's no blood-brain barrier involved there. And so
because it's like a electrical.
Well, it's a cell. It's an actual cell. It's actually physically connected. It's physically connected. It's not electrically connected. It's physically connected. So you have an axon with a terminal field, like the terminal boutons that are connecting right to the gut. They're sensing the gut, right? And so anything that gets into that terminal field, the terminal synapse, okay. So when your gut microbiome is emitting nasty stuff, like things that are neurologically toxic, for example, okay, it's no different than getting a toxin in your brain that gives you Parkinson's, right? So it's actually going to affect that synapse, and it's going to, and in some ways, it can actually go in the neuron, and it can go up the neuron axon all the way into your brain. Like you can actually have molecules that go right from the gut into the brain, okay, without passing through the gut blood supply at all, because that's a physical cell connecting the two.
Wow.
Okay, and so likewise in the afferent system, you'll have the axon now is, instead of going from the brain to the gut, the axon is going from the gut to the brain, and so it's going to go up there, and it's going to make some signals to things, right? And so this will be like if you have distension or things like that can affect it. And then the third neurological system is the connection between the two, efferent and afferent. There's a what's called an enteral neurological system, which is mostly the serotonergic system. When people talk about serotonin in the gut.
Yeah, like 95% of your serotonin apparently is
in
your
gut.
That's
the peristaltic type motion.
So the serotonin is made in your gut and then sent to your brain?
No, it's made in your gut, but the serotonin stimulates the neurons that are connected. And so it doesn't actually go to the brain, other than through the blood supply, but it'll actually, the enteral neurons will either stimulate or suppress the other neurons going back and forth.
Wow. And that's why they say, correcting a dysbiosis in the gut can help with even mental health.
Oh, absolutely.
Yeah. Wow.
Because it's affecting that connection. And so, and if you think about it, anything that changes your neurological structure, like, so if I, poke you with the stick, right, you're going to have a pain sensor, and you're going to feel that pain in your brain, right? So fundamentally, your gut is no different. Like if your gut is part of your environmental stimulus. So just like anything in our environment that we do, okay, whether I see a happy face or a sad face can actually give me depression or anxiety in my brain, because I'm sensing those type of interactions. So it is connected in exactly that way.
That's so interesting. What you said earlier about being relaxed versus you eat a big meal and someone comes in with a gun, it reminded me of I was having digestive problems and I went to see a specialist. It was very expensive and very hard to get into and blah, blah, blah. And after my big expensive appointment, he told me to sit down and to chew my food and to eat slowly. And those were like his major takeaways. Yes. But I think that actually probably holds true.
Yeah. Well, it really does hold true. And I think back to the psychology of your gut, which is the psychology of eating, right? Like we do need to eat, and we need to make that into a pleasant experience where you've taken it calmly. Your digestion has time to work. It works in its proper time and place. Those are all very positive things. Absolutely.
So what happens when when you have a dysbiosis or the gut is not in balance? What are some of the symptoms or the conditions or things that can result from that?
So typically when you're talking about gut dysbiosis, or there's two aspects of it. So the dysbiosis part is the microbiome being disrupted in certain areas of your gut. But the gut dysfunction from an inflammatory perspective means a leaky gut. Okay, so your gut is supposed to be your barrier. A leaky gut is analogous to atherosclerosis, for example. So we talk about atherosclerosis where you have the endothelial lining of your vascular walls. It gets gaps, and molecules get in between the gaps, and they create inflammation in this wall, which causes
your arteries.
Of the arteries.
Okay.
Which is fundamentally atherosclerosis. Same thing happens in the gut. So what happens is the cell-to-cell enteral walls become leaky.
So when you were talking earlier about how it's a closed system.
Correct.
Leaky gut is this closed system is no longer closed.
Correct. Now, and it leaks into the gut. If you take a look at it, it's very, very highly folded, if you will, right? It's not
kind of like the brain.
Yeah, it's not like the brain, exactly. It is, and it's whereas like the arteries and the vasculars are much more smooth and straight, but you have these concentric circles of layers, if you will, and when they break through a certain layer, and it's really interesting because when you digest your food, it goes through cell layers. It's kind of cool because it's like inside is the lumen and the liquid, if you will, and as it gets digested, the molecules kind of go through these cells and get reattached, and they get into your blood supply and into your liver. Anyway, so the point is when the first line of the first cell membrane wall, if you will, gets inflamed, gaps can occur in between the cells. And then actual material, rather than just metabolites from biochemistry going through the standard membrane, you actually get particles or bacteria. You actually have proteins and things that actually
going into your blood.
Into your blood, or if they go to the blood, it's really bad. So blood, if it goes into your blood, you have sepsis. That's bad. Okay.
Okay.
So typically, the first part is inflammation. It goes through, and you get this leaky gut, and it's going to create these huge pro-inflammatory mediators. It's like the body is recognizing this as bad and is trying to find a way to fight it. And so the leaky gut creates all this inflammation. So now you got all these pro-inflammatory mediators, which creates anxiety. It's highly linked to autism and inflammatory brain inflammation diseases. So it's truly an inflammatory engine, if you will. I don't think you have to go beyond that.
And then you're probably not even processing healthy food well.
Correct.
Like it's like this snowball effect where you're trying to eat healthier food and take supplements and things, but if your gut wall if it's leaky, then it's probably not processing things as good as it could be.
Correct. And it doesn't happen all in the same place. So people talk about the difference between irritable bowel syndrome and inflammatory bowel syndrome. Okay, so an inflammatory bowel syndrome is people that have really inflamed bowels. You can get blood in your stool, and so you're actually your intestinal walls are so inflamed that they're bleeding. They actually look like a cut hand, if you will. They're just like with 1,000 little cuts, right? That's inflammatory bowel syndrome, and that inflammation can ultimately lead to something like cancer. Then you have what's called irritable bowel syndrome. Now, irritable bowel syndrome is much more common, where people have bouts of diarrhea followed by bouts of constipation. And people, so what happens in that situation, you have areas of this very, very long tube that get disrupted. And so irritable bowel syndrome typically is a situation where you'll have pockets in the gut that become dysregulated.
Irritated?
Yeah, and it's almost like a hernia, if you will, right? You get these little bulges. And so when we're trying to fix that, this is a good example where people take a laxative or fiber and they try to get, if they're constipated, for example, and they try to get rid of constipation is a big deal. So when we're talking about health, anything optimizing health, you really should start with the gut. Like fundamentally, people need to be having proper gut function. Like constipation in a child, constipation in an adult, constipation is just bad news all the time. So you need to be pooping every day. Okay, that's just rule number one. You got to get your gut mobility properly functioning. But what happens with people with irritable bowel syndrome is that they'll have constipation, and when you get constipation or compactation, the gut walls are a tube, a flexible tube, and they flex up and down as you eat and as you have fluids in them. But what will happen is when you get compacted, it gets distended, and then when you have like a fiber or metamucil or Miralax or some sort of, or even lactulose, something that'll loosen the gut, hydrate the gut, and you'll evacuate. The gut walls don't immediately relax back to normal again. So you're stuck with this distended wall. And if they don't continue, so what happens then, you get this diarrhea that dumps everything out. And then what happens is it starts filling up again, and it fills up in these little holes again, these little distended areas. And so then you end up with constipation, then diarrhea, then constipation, then diarrhea. So one of the biggest things that people don't do properly is when they get on a laxative, is that they say, "Oh, I'm normal now, I'm going to stop my laxative." You want to continue that and slowly taper it off because you need time for that gut wall to actually relax, come back down. Otherwise, you're going to constantly have this, what's called irritable bowel syndrome. And so when we're talking about restoring gut function fundamentally is about restoring the biochemistry of the gut, restoring the leaky, like there's that inflammation and the leaky gut aspect of it. And then there is the gut microbiome distribution. And those things are actually not that hard to do overall. So it depends on how severe the individual is.
Yeah. So it's the structural, like the function of the actual gut, the tube. Right. Is that structure, is that system healthy? Yeah. And then it's also the health of the microbiome that lives within that system needs to be in balance. So correct. It's a different approach. So back to what we started our conversation with, which is these prodromes.
Right.
So when you took the cancer out and the person was cancer-free, they still had these prodromes of an imbalance.
Correct.
So that's hinting at the underlying things, whether it's the health of the system of the gut or the microbiome, still needed to be addressed.
Correct.
So how do you heal the walls of gut lining? How do you heal a leaky gut?
So there's really three things about maintaining or restoring gut function. On the most severe side, there's like fecal replacement therapy, right, where you can actually restore the gut flora with someone's healthy gut flora, right? Like, so you can actually do, enema of. I've
heard of this fecal transplants. Exactly what it sounds like.
And so that's on the extreme side, but there are a lot of really good success with it. And it's not new. They've been doing it since the 50s or 1800s. Even. Actually, in fact, it started with husbandry in animals, veterinary medicine. And so, ironically enough, like, so you'd have people managing horses. So if they had a couple horses in the field, and this is was just at a gut microbiome conference, and I can't remember when the speaker was telling me the story, but I thought, wow, that's amazing. And so what they do is someone would have one horse that have bad diarrhea and one horse that would be normal. And so what they would basically do, and especially if they would basically reach into the healthy horse butt, okay, and then basically take that arm sleeve and put their arm sleeve into the unhealthy horse.
My goodness.
And in no time, the unhealthy horse had no diarrhea.
So if you zoom out, they're really repopulating.
Yeah.
So if that wasn't an option, you could repopulate with just, fermented foods and probiotics and prebiotics.
So that's the severe case. So let's talk about what most everyone can do.
Whoa.
So obviously I deal with this a lot in our clientele.
Yeah.
And the rule of thumb is really three things. One is you want to first and foremost make sure you seal a leaky gut, and there are the plasmalogens are critical for that. There's some peptides that work. Like there's an oral peptide called often called BPC 157. It's available as an oral supplement. For example, it's a peptide that your stomach makes, and it's been pretty good at reducing leaky gut syndrome. And then certain curcuminoids are very powerful in terms of anti-inflammatory.
Because they're anti-inflammatory.
Right. And very specific ones that have very similar activity to these natural GTAs. And so those
What does GTA stand for again?
Gastrointestinal tract acids. So
it's literally the acid inside your gut.
They're made by gut microbiomes. These are the biomarkers that we use.
We talked about these earlier.
Exactly.
So the microbiome makes these.
Correct.
Okay.
But they have the most similar nutritional supplement to those molecules are the curcuminoids, and specifically the third curcuminoid. There's three curcuminoids, 1, 2, and 3. The third one is called bis dimethoxycurcumin, so BDMC, and it has very, very powerful anti-inflammatory activities in the gut. It actually stimulates a specific microRNA called microRNA 148A. If you want to get totally geeky, and it'll actually help seal the gut. So the inflammation. You really want to make sure you've resolved the inflammation of the gut, and you can deal with the inflammation of the gut independent of the microbiome, because
that's like structural problem.
It's biochemistry. You're actually you're feeding the enterocytes, the actual cells of your gut wall, with anti-inflammatory molecules and nutrition that they use independent of your gut. And then secondly, in terms of the gut itself, is you want to make sure that you're, like where you said with the fermented foods are wonderful.
And you want them to be refrigerated. Like even if you're taking, if something's going to have live active cultures, if it's not in the fridge, it's probably not so alive.
Mm-hmm. Well, it's interesting in terms of the whole probiotics.
Yeah.
It's kind of a very complex field, and it's pretty hit and miss. I think the challenge with a probiotic is designed to actually inoculate the gut with new bacteria. Okay.
So the actual probiotic contain microbiota, like the actual bugs.
Correct. And so
the
goal is and the trick is, can you get them through your mouth and your stomach, and can you get them into the location of the gut that you need them? And then will they actually be incorporated is the second thing. Because remember, it's a community, right? And you're sending in
might
these...
be kicked out. If it's all of the bad bugs, they might say, no, thank you.
No, thank you. So that's a probiotic. And that's why, and fundamentally, the fecal replacement is a probiotic strategy. It's just, it's delivering them through
So
an
if
animal.
you had to choose personally between a probiotic and a prebiotic, would you actually go with the prebiotic?
I think the prebiotics is much more reliable. And that's where the fermented foods come in. Because what you're doing then, assuming that it's unlikely that you have completely nothing of good bacteria. And so by changing your diet and changing the biochemistry of your body, okay, your gut microbiome will adjust accordingly. Vitamin C preferentially improves bifidobacteria.
Interesting.
There's certain immunoglobulins that will actually help. So there is prebiotics that will selectively enhance one type. And if you do a combination of prebiotics plus gut-focused biochemical restoration, then the gut will gradually adjust to that new opportunity, that new system.
Yeah.
And that's the biggest thing.
There was a really interesting study out of Stanford that had people on high fiber diets versus high fermented food diets. And the fermented food diet, their inflammation markers were way down in their gut, and the microbiome was actually more diverse. Like
Yes.
it stuck, it worked. And so, but it's kind of like by doing the prebiotic route, you're kind of fanning the flame of the good ones.
Right.
You're like allowing those ones to.
And exactly. But see, the reason why fermented foods work is because the last thing I forgot to mention to everybody is that the microbiome is a community, and it's mostly an anaerobic community, and that these different bacterias, some produce molecules that are food for others. So it's not just they themselves, the gut microbiome is not just in symbiosis with the human host. They're in symbiosis with each other, right? So good bacteria will actually create molecules that other bacteria use as food supply. And so when you are eating a fermented food, what you're doing is you're actually eating microbiome products. Like, so you're eating molecules, like there's the biochemistry of a fermented food has been created by microbes, which is the definition of fermentation. And so what you're doing is the fermentation process itself is creating prebiotic biochemicals that are preferentially used by gut microbiome. And so that's the whole. So it's the same concept as the lactulose, which selectively will enhance the bifidobacteria and lactobacillus. The fermented foods you're actually giving yourself healthy bacteria food.
It's like fertilizer, kind of?
Exactly.
Wow.
Yeah. But that's the fertilizer that fertilizes just the crop, not the weeds.
I mean, I'm about to go buy some yogurt when we're done, honestly, some kefir. That's so interesting. So to kind of wrap it all up with, what you were talking about before with prodromes, could someone use a blood test to maybe see if they do need a colonoscopy? Like, can you use like looking at things on a blood test to figure out how your gut is functioning?
Absolutely. Because your functioning of your gut, the entire purpose of your gut microbiome is biochemical. These are organisms. These are bacterial organisms. They do not float into your blood supply. Okay, so their interaction with the human host is entirely biochemical. So the relationship between humans and their gut microbiome is all related to the biochemical interaction between them and our blood supply, period. So our blood supply, if you're diabetic or if you have, if there's something wrong with your liver or other things, your microbiome will actually adapt. So it's a biosensor of the human body as well. But fundamentally, it's making biochemicals that are either being absorbed into your blood supply, which can be inflammatory. They could be absorbing, they could be influencing your neurological system because they're either stimulating neurons or they're being absorbed by neurons and having toxic results. It's all biochemical by definition. It is a fermented vat of microorganisms, and it's no different than you making beer or wine. And you can either have sweet tasting wine or you can have vinegar based upon the composition of the microorganisms and the food, the foodstuffs that you are using to feed it. It is actually no more complicated than that. And so if you want, at the end of the day, it becomes two things. What is the distribution of the microbiome and what are you feeding it? And those two things will determine the microbiome component of it. And then secondarily is your human, the host, the enterocyte walls.
The structure.
The structure.
Yeah.
Because there's your biochemistry, remember. So the microbiome is not building your gut wall. Your liver and your body is building the gut wall. Okay, that's you. So the cells of your gut are made by you. They're maintained by you. And so the plasmalogens, the B vitamins, the methyltransferase system, mitochondrial support structure, all of that stuff, that's on you. Okay, it's on you to have a healthy gut wall, if you will. That's not the microbiome can work against you or for you, but it's not. It's an environmental stressor on it. The microbiome, on a second perspective, is so they interact, but they are two different things.
And so, if you did blood work, what would you maybe see if your gut health wasn't ideal?
Well, a couple of really important ones. So there's the short chain fatty acids, like butyrate, propionate, acetate can be measured.
Would you see high level, low levels of those?
Well, actually, high levels are usually pretty good of the butyrate.
Okay.
Propionate is typically considered bad, pro-inflammatory. These gastrointestinal tract acids are a big, powerful marker because they're so linked to different disease states. And then there's another one that people are very common with. It's called TMAO, so trimethylamine oxide. And so people think, so high levels of TMAO are linked to cardiovascular disease. And people say, oh, then you shouldn't eat these choline-containing foods, because if you eat choline-containing foods, you can increase these TMAO levels in your blood. But in fact, TMAO levels are an indicator of bad gut health.
Interesting.
Because actually TMAO itself is protective of your heart. So this elevated TMAO is usually an indicator that you have bad gut health. So your gut is also, like you talk about Parkinson's and Alzheimer's disease, like so the amyloid system or the alpha-synuclein system in Parkinson's. So the gut is a huge component of human physiology, even though it's not in our body. And so all that comes, so it's all biochemical. So the bottom line is yes. So these GTAs, the other big ones for colon, pancreatic is phosphatidylcholine.
Interesting.
So low levels of phosphatidylcholine, low levels of plasmalogens, low levels of GTAs. These are three really robust indicators of a prodrome of future gastrointestinal disease states.
That's so interesting. And maybe you could use those to then see if you need to intervene or the colonoscopy.
Correct. And so when we did these large studies, so we've run 7 colon cancer trials, right?
Wow.
And so the very large colonoscopy trial, one of the largest done in Canada, with just under 5,000 colonoscopies and about 1,000 individuals in the general population. And these are people that are going in for colonoscopy, high-risk individuals. So they're already having some sort of GI symptoms. And we were trying to find out, can we better triage individuals as to their cancer risk, the risk of polyps transitioning into cancer? And the answer was yes. With a simple blood test, we were able to diagnose or identify 87% of all the colon cancers.
Wow.
Okay, with the same blood test that we use for inborn errors of metabolism, or same technology, if you will. And then we could actually identify people who had polyps, high-risk polyps versus low-risk polyps, because one of the issues with removing polyps is that they can bleed and things like that. So the answer is yes. So we can, from a mathematical perspective, identify people at high risk versus low risk. And so people that had normal GTA levels had extremely low risk of colon cancer. And people with high GTA with low GTAs were high risk. So then you would recommend those individuals to go in for colonoscopy. Same thing with pancreatic cancer. Biomarkers in the blood indicating that pancreatic cancer or pancreatic function is impaired. That would indicate this is an individual that should go in for an ultrasound. Because the biggest thing with pancreatic cancer is it's asymptomatic for so long. Unless you're lucky or unluckily, as you want to call it, that you actually have a bile duct being blocked and you get some sort of,
signal
like hepatitis or bilirubin, like you get some sort of yellowing, right? That would indicate that something is blocked. You go undiagnosed. So when people get diagnosed with pancreatic cancer, it's usually very late. Same thing with ovarian cancer, for women. So the biochemistry is extremely powerful at understanding human physiology. What is the physiological state of the organism at this point in time? Is it in a healthy state? Is it in has it deviated from health? And it has that, and the deviation from health that you might see in an individual, that deviation, can you predict what health outcome would occur from that particular deviation? Right? We talk about plasmalogens and its association with dementia, Okay, is associated with cancers. GTAs are more associated with cancers, and so there are certain deviations from health that have a higher association with some certain disease endpoint. And so
it's so interesting every time we speak that it feels like you're any topic you seem to know so much about it. You're an expert in everything, but really it's because this biochemistry is universal.
It's universal. And I've been doing this for a very long time.
Yeah, that too.
Okay. You go through hundreds of thousands of blood samples across the world.
Wow.
And these things they happen. And that's why that's why this whole concept of restorative health, right? Where we ignore the disease state and say, you know what, let's forget disease. The human body has an operating specifications that it's designed to run under. Let's just restore the operating condition as it's designed to be run and support that. And this whole restorative health comes from the prodrome. So the natural progression from a prodrome is saying, okay, how do I restore a prodrome to health? And then that leads us back to the gut again, because we have to supply these molecules. It's now what vitamins, what nutrients, at what time of day, how do we deliver to the human body, to this human bioreactor that we all have? How do we make sure we have the right material in the right place?
Yeah, and the gut is just foundational. It's the one that's processing, breaking things down, and sending them where they need to go.
And so that's when we design the molecules, right? These plasmalogen precursors are designed for our gut our gut metabolism to metabolize and absorb rapidly in the upper GI.
Because they can make it through the stomach acid, whereas if you're eating it in your food, those plasmalogens can't. So interesting how you can apply this approach to so many different things. Yeah.
It really is. I think we overcomplicate on some areas and oversimplify in others, but fundamentally it's like I tell people, like, you eat a hamburger and a milkshake and your body you make hair with that. Right, you make you make your lungs with that. You make heart with that. Like, think about that. You can't think of a more advanced, bioconversion system than the human body.
Yeah, every time we chat, it's just more and more miraculous to me. So thank you so much for joining me today. This was wildly fascinating. I learned so much, and I will never underestimate the power of my gut health again after this. So thank you.
Well, you're welcome, Victoria. It was a pleasure.